Imagine a world shrouded in the constant dread of infection, where a simple scratch could lead to agonizing death, where childbirth was a gamble against puerperal fever, and where even a minor illness could swiftly escalate into a fatal struggle against unseen invaders. This was the grim reality for millennia, a relentless war waged by humanity against microscopic foes, often with little more than prayer, folk remedies, or the crude surgeon's knife as their only weapons. The annals of history are filled with countless lives extinguished prematurely, not by grand battles or catastrophic natural disasters, but by the silent, insidious creep of bacterial infections that laid waste to populations, decimated armies, and brought even the mightiest individuals to their knees. Civilizations rose and fell, empires expanded and contracted, yet the microscopic world remained an unchallenged sovereign, dictating the very terms of human existence. For centuries, medical practice, though evolving, remained largely impotent against the scourge of infectious disease. Physicians, with their limited understanding, could only observe and, at best, offer palliative care, their efforts often akin to bailing water from a sinking ship with a thimble. The very air seemed to harbor unseen threats, and the simplest wound became a portal for an agonizing demise. The notion of a true cure for these ubiquitous ailments seemed a distant, unattainable dream, confined to the realm of alchemists' fantasies or the whispered promises of charlatans. Yet, amidst this pervasive despair, a whisper of hope, seemingly plucked from the ether, began to emerge from the most unexpected of places. It was not the culmination of centuries of arduous research, nor the result of a deliberate, grand scientific expedition. Instead, it was an accident, a serendipitous alignment of circumstances, a moment of extraordinary observation by an ordinary man, that would forever alter the course of human history. This is the story of that pivotal moment, of a forgotten petri dish, a curious scientist, and the unassuming mold that would become one of humanity's greatest saviors. It is a testament to the power of keen observation, the relentless pursuit of knowledge, and the profound impact of what can seem, at first glance, to be a mere oversight. This narrative will journey through the fascinating journey of penicillin, from its humble origins in a London laboratory to its monumental role in transforming medicine and redefining the very boundaries of human life and longevity. It is a story not just of scientific triumph, but of the remarkable human spirit, the tireless dedication of researchers, and the enduring legacy of a discovery that continues to resonate profoundly in our modern world, where the fight against disease remains an ongoing endeavor. Prepare to delve into the fascinating tale of a discovery that was both utterly accidental and profoundly transformative, a discovery that gifted humanity a shield against unseen enemies and ushered in an era of unprecedented health and progress. Before the dawn of antibiotics, the world lived under a pervasive shadow of fear, where the simplest cut could lead to an agonizing death, and diseases now easily treatable were once devastating scourges. Imagine a time when hospitals, far from being havens of healing, were often places where infections spread rampantly, where the stench of gangrene hung heavy in the air, and surgical procedures, though necessary, carried an almost insurmountable risk of post-operative infection. The concept of microscopic life, though theorized by a few visionary minds, was largely misunderstood, and the true mechanisms of disease transmission remained an enigma to most medical practitioners. For centuries, treatments were rudimentary at best, ranging from herbal concoctions to bloodletting, methods often more harmful than helpful, rooted in ancient superstitions and limited empirical observation. Childbirth, a joyous occasion in modern times, was then a perilous journey, with puerperal fever claiming the lives of countless mothers, leaving families shattered and communities grieving. Even seemingly innocuous childhood illnesses like measles or scarlet fever, now largely contained by vaccines and antibiotics, could sweep through communities like wildfire, leaving a trail of death and disability in their wake. Tuberculosis, a relentless and insidious killer, silently ravaged populations, lingering for years in its victims, slowly consuming them from within, a fate almost universally dreaded. Pneumonia, a common respiratory infection, was often a death sentence, especially for the elderly or infirm, their lungs filling with fluid as their bodies succumbed to the relentless bacterial assault. Surgical advancements, though slowly progressing, were severely hampered by the ever-present threat of infection. A successful operation might be undone by a subsequent bacterial invasion, rendering the surgeon's skill moot. The very concept of sterilization was in its nascent stages, and antiseptic practices, championed by pioneers like Joseph Lister, were met with skepticism and resistance from a medical establishment steeped in tradition. Patients often faced amputations to prevent the spread of gangrene, enduring excruciating pain with little effective anesthesia, knowing that even then, survival was far from guaranteed. War, already a horrific endeavor, became even more deadly due to infected wounds, with more soldiers dying from disease than from direct combat, a grim testament to the unchecked power of bacteria on the battlefield. Families lived with the constant fear of losing loved ones to illnesses that today are mere inconveniences, and entire communities could be decimated by outbreaks of diseases like cholera or typhoid, which spread rapidly through contaminated water and inadequate sanitation. The average life expectancy was significantly lower, a stark reflection of humanity's vulnerability to microbial threats, and the concept of a long, healthy life was often a luxury reserved for the exceptionally fortunate or resilient. Doctors, despite their dedication and compassion, often found themselves helpless, their hands tied by the limitations of their knowledge and the absence of effective tools to combat the invisible enemies that preyed upon humanity. This was the world that awaited a discovery, a world desperately in need of a breakthrough, a world on the cusp of a revolutionary change that would redefine health, medicine, and the very parameters of human existence. The stage was set for an accidental hero, a quiet observer whose seemingly insignificant discovery would shatter the chains of microbial fear and usher in an era of unprecedented medical advancement and hope. In the quiet, unassuming environs of St. Mary's Hospital in London, amidst the bustling wards and the constant hum of medical activity, resided a man whose meticulous observations and inquisitive mind would inadvertently alter the course of human history: Alexander Fleming. Born in rural Scotland in 1881, Fleming's early life on a farm instilled in him a deep appreciation for nature's subtle intricacies and a keen eye for detail, qualities that would prove invaluable in his scientific pursuits. He was not a flamboyant figure, nor one to seek the limelight. Rather, he possessed a quiet, introspective demeanor, a methodical approach to his work, and an almost childlike curiosity about the microscopic world. His laboratory, a small, somewhat disheveled space, was a reflection of his personality. A place of focused inquiry, where experiments often overlapped and unexpected results were not dismissed but meticulously examined. Fleming's primary field was bacteriology, a relatively new discipline at the turn of the 20th century, dedicated to understanding the role of bacteria in disease and finding ways to combat them. He had a particular interest in antiseptics, having served as a captain in the Royal Army Medical Corps during World War I, where he witnessed firsthand the horrific toll of infected wounds on soldiers, a sight that profoundly affected him and fueled his desire to find better solutions. He was frustrated by the ineffectiveness of many existing antiseptics, which often killed the body's own immune cells along with the bacteria, making the patient worse rather than better. This experience honed his focus on finding substances that could specifically target harmful bacteria without damaging human tissues. His daily routine involved cultivating various bacterial cultures, examining them under the microscope, and carefully documenting their growth and behavior, a seemingly mundane task that required immense patience and precision. The laboratory itself was a hub of activity, filled with petri dishes, test tubes, and the faint, distinct smell of bacterial cultures, a familiar aroma to anyone working in the burgeoning field of microbiology. Fleming's colleagues recognized his quiet brilliance and his unwavering dedication to his research, even if his methods sometimes appeared unconventional or his workspace somewhat untidy to the casual observer. He was known for his sharp intellect and his ability to synthesize disparate pieces of information, often seeing connections where others saw only isolated facts. His laboratory was more than just a place of work. It was a sanctuary of discovery, where the boundaries of scientific knowledge were constantly being pushed, sometimes through deliberate experimentation, and sometimes, as fate would have it, through pure serendipity. The atmosphere was one of quiet contemplation punctuated by moments of intense focus, where the pursuit of understanding the invisible world of microbes was paramount. Fleming's life was characterized by a relentless pursuit of answers, a deep-seated desire to alleviate suffering, and an unwavering commitment to the scientific method, even when faced with setbacks or skepticism. It was within this specific context, this particular laboratory, and under the watchful eye of this dedicated bacteriologist, that the threads of fate would converge to create one of the most significant medical breakthroughs in human history. His story is a powerful reminder that profound discoveries often emerge not from grand pronouncements, but from persistent, humble inquiry and the readiness to recognize the extraordinary in the seemingly ordinary. The summer of 1928 arrived in London with its usual tempered embrace, prompting many to seek respite from the city's confines. For Alexander Fleming, a much-needed vacation awaited, a customary escape from the meticulous demands of his bacteriology laboratory at St. Mary's Hospital. In the days leading up to his departure, Fleming, like any scientist, was engaged in his routine work, preparing and examining various bacterial cultures, including those of Staphylococcus aureus, a common bacterium known for causing a range of infections, from boils to life-threatening sepsis. He was known for his somewhat untidy lab habits. Petri dishes often remained stacked on his workbench for longer than was ideal, a testament to his focused, if occasionally disorganized, approach to research. As he tidied up, or rather, as he prepared to untidy himself from the laboratory for his holiday, some of these bacterial cultures were left inadvertently uncovered, or perhaps simply pushed aside, rather than being properly disposed of or meticulously stored away. The London air, carrying with it a myriad of microscopic life forms, drifted through the open windows of his laboratory, a silent, unseen current carrying spores and dust particles from the outside world into the scientific sanctuary within. Among these airborne travelers were spores of a common mold, Penicillium notatum, a fungus typically found in damp environments, on decaying organic matter, or even on stale bread. One of these microscopic spores, carried by a gentle breeze or perhaps deposited by a chance air current, found its way onto one of Fleming's neglected Staphylococcus culture plates. The conditions within the laboratory during Fleming's absence, the ambient temperature, the humidity, and the time allowed for incubation, were, by sheer happenstance, precisely favorable for the mold spore to germinate and begin to grow. While Fleming enjoyed his time away, perhaps strolling through the Scottish countryside or simply enjoying a quiet retreat, an invisible drama was unfolding back in his London laboratory. The Penicillium mold began to spread across the agar surface of the Petri dish, slowly but inexorably, its filamentous hyphae reaching outwards. As it grew, it began to secrete a substance, a metabolic byproduct, into its immediate environment, a chemical compound that would soon prove to be of immense significance. This secretion diffused through the agar, creating a clear, bacteria-free zone around the growing mold colony. The Staphylococcus bacteria, thriving elsewhere on the plate, were mysteriously inhibited and destroyed in the vicinity of the mold, creating a striking visual contrast, a vibrant green mold colony surrounded by a halo of emptiness, while the rest of the plate teemed with bacterial growth. This unnoticed biochemical warfare continued for several weeks, a silent testament to nature's ongoing microscopic battles, all occurring within the confines of a forgotten Petri dish. The stage was set for a discovery, not through deliberate intent or meticulous planning, but through a confluence of circumstances, a scientist's holiday, an open window, a stray mold spore, and the unseen processes of microbial competition. The summer of 1928, otherwise unremarkable, would forever be etched into the annals of medical history as the period when the seeds of a revolution were sown, not in a grand experiment, but in a moment of accidental neglect. The return of Alexander Fleming would soon unveil this silent revelation, transforming a seemingly trivial oversight into a monumental turning point for humanity's fight against disease. Upon his return from vacation in early September 1928, Alexander Fleming surveyed his laboratory, a scene of organized chaos familiar to any dedicated researcher. As he began the arduous task of clearing and cleaning his workbench, sorting through stacks of petri dishes, a particular plate caught his eye. It was one of the Staphylococcus cultures he had prepared before his departure, and it was strikingly different from the others. Instead of a uniform carpet of bacterial growth, a distinct patch of green mold had colonized a portion of the agar. What truly arrested his attention, however, was not merely the presence of the mold, but the remarkable absence of bacterial colonies immediately surrounding it. A clear, circular zone, almost like a perfect halo, separated the flourishing mold from the otherwise robust growth of Staphylococcus bacteria across the rest of the plate. Many scientists, upon encountering such a contaminated plate, would have simply discarded it, dismissing it as a ruined experiment. But Fleming was different. His keen observational skills, honed over years of meticulous bacteriological work, immediately recognized the anomaly as something potentially significant, not a nuisance, but a clue. He peered closer, his curiosity piqued, his mind already racing with hypotheses about this curious phenomenon. He realized that whatever the mold was producing, it was having a profound inhibitory effect on the bacteria. This was not a random occurrence. There was a direct, localized interaction that demanded further investigation. He carefully transferred a sample of the mold to a fresh culture plate, initiating a pure culture of the fungus for detailed study. He also took samples from the clear zone and the bacterial growth areas, meticulously preparing slides for microscopic examination. Under the microscope, he observed that the bacteria within the clear zone were either dead or had failed to grow, while those further away were perfectly healthy. He identified the mold as a species of Penicillium, later confirmed to be Penicillium notatum, a common contaminant, yet one that, in this specific instance, held an extraordinary secret. This moment was not just one of discovery, but of profound insight, the ability to see beyond the superficial contamination and grasp the underlying biological significance. Fleming's mind, always questioning, immediately considered the potential implications. If this substance could kill bacteria in a petri dish, could it also do so within the human body, without harming human cells? He began to refer to the active substance produced by the mold as mold juice, and later, more formally, as penicillin, after the genus of the mold. He conducted further experiments, testing the mold juice against various other pathogenic bacteria, including those responsible for scarlet fever, pneumonia, and diphtheria, consistently observing the same inhibitory effect. Crucially, he also tested its toxicity to human white blood cells, the body's natural defense mechanisms, finding that it was remarkably non-toxic, a critical distinction from the harsh antiseptics he had previously criticized. This initial serendipitous observation, combined with Fleming's astute scientific mind and persistent experimentation, laid the foundational cornerstone for what would become one of the most transformative medical breakthroughs in history. It was a true aha moment, born from a blend of accident, careful observation, and the profound ability to connect disparate pieces of evidence into a revolutionary concept. The journey from this petri dish to widespread medical use would be long and arduous, but the first, indispensable step had been taken, forever changing the trajectory of human health and our understanding of microbial warfare. Following his remarkable observation, Alexander Fleming embarked on a series of rigorous experiments to understand the properties and potential of his newfound mold juice, which he named penicillin. He meticulously grew cultures of Penicillium notatum, extracting the liquid in which the mold had grown, and then tested its antibacterial properties against a wide spectrum of pathogenic bacteria. His results were consistently astonishing. Penicillin demonstrated potent bactericidal effects against many of the most virulent bacteria responsible for widespread diseases, including streptococci, staphylococci, and diphtheria bacilli. Crucially, his experiments also showed that penicillin was remarkably non-toxic to human cells and animals, a critical advantage over the existing antiseptics of the time, which often harmed the host along with the pathogen. He published his findings in 1929 in the British Journal of Experimental Pathology, detailing his observations and suggesting the potential therapeutic utility of penicillin as an antiseptic. Despite the groundbreaking nature of his discovery, Fleming encountered a significant amount of skepticism and indifference from the wider scientific and medical community. This lukewarm reception stemmed from several factors. Penicillin was difficult to isolate and purify in stable quantities, making it challenging to produce enough for clinical trials. The active substance seemed to degrade quickly, losing its potency, which presented a formidable obstacle to its practical application. Furthermore, the prevailing scientific focus at the time was on synthetic chemical compounds for drug development, not on substances derived from molds, which were often seen as unreliable and unpredictable. Break time equals 0. 25s / the medical community, accustomed to the limitations of existing treatments and perhaps jaded by past false dawns, was slow to grasp the revolutionary potential of Fleming's discovery. Some colleagues dismissed it as merely another antibacterial agent of limited practical value, while others found the concept of a mold juice as a therapeutic agent somewhat unscientific or even fantastical. Fleming himself, though convinced of penicillin's power, was primarily a bacteriologist, not a chemist, and he lacked the expertise and resources to effectively purify and mass-produce the compound. He continued to use crude penicillin extracts in his laboratory for various purposes, such as selectively isolating bacteria or in some limited topical applications, but he struggled to develop it into a stable, widely usable drug for systemic infections. His initial attempts to generate interest in large-scale production or further chemical refinement were met with polite but firm rejections from pharmaceutical companies and research institutions. For over a decade, penicillin remained largely a laboratory curiosity, its immense potential slumbering, awaiting the right combination of scientific expertise, persistent effort, and a global crisis to ignite its widespread development. The world, still reeling from infectious diseases, was unknowingly sitting on the precipice of a medical revolution. Yet the key to unlocking it remained largely unappreciated, a testament to the fact that groundbreaking discoveries often require more than just initial insight. They demand perseverance, validation, and the confluence of further scientific advancements to truly realize their transformative power. Fleming's invaluable contribution was to identify the phenomenon and highlight its potential, but the monumental task of transforming a laboratory curiosity into a life-saving medicine would fall to others. While Alexander Fleming's initial discovery of penicillin was a monumental first step, the chemical and medical world largely overlooked its profound implications for over a decade. The arduous task of transforming Fleming's unstable laboratory curiosity into a life-saving, mass-producible drug fell to a dedicated team of scientists at the University of Oxford, working under the demanding yet visionary leadership of Howard Florey. Florey, an Australian pathologist and physiologist, possessed a relentless drive and an unwavering commitment to translating scientific research into practical medical applications. He was not easily deterred by previous failures or the perceived limitations of a project. In 1938, intrigued by Fleming's decade-old paper, Florey decided to dedicate the resources of his pathology department to systematically investigate naturally occurring antibacterial substances. Central to his team was Ernst Chain, a brilliant German-born biochemist with an exceptionally sharp mind for chemical analysis and purification. Chain, who had fled Nazi Germany, brought with him an unparalleled expertise in organic chemistry and enzyme action, skills that would prove indispensable in unraveling the elusive structure of penicillin. His methodical approach to biochemical extraction and purification was precisely what was needed to tackle the challenges of isolating penicillin in a stable form. The third crucial member of this pioneering trio was Norman Heatley, a gifted and ingenious biochemist, whose practical skills and inventive solutions to experimental problems were legendary within the team. Heatley was the master of improvisation, designing and building the complex apparatus required to grow the mold in large quantities and extract the fragile penicillin, often creating ingenious contraptions from everyday laboratory materials. His ability to overcome engineering and scaling challenges was vital to the project's success, transforming theoretical concepts into tangible processes. Together, this interdisciplinary team embarked on a painstaking journey to grow the penicillium mold in large quantities, isolate the active compound, and test its efficacy and safety in living organisms. Their laboratory in Oxford became a hive of intense activity, fueled by long hours, meticulous experimentation, and a shared vision of unlocking penicillin's full therapeutic potential. They refined methods for culturing the mold, discovering that certain conditions maximized penicillin production, and developed sophisticated chemical techniques to extract and purify the incredibly unstable molecule. The initial quantities they could produce were minuscule, but each precious milligram was meticulously tested, first on mice, then on increasingly larger animals. Their experiments on mice, infected with lethal doses of streptococci, yielded astonishing results. The mice treated with penicillin survived, while the untreated control group succumbed to the infection. These animal trials provided irrefutable proof of penicillin's efficacy in vivo, a critical step that had largely eluded Fleming. The Oxford team's work was characterized by an extraordinary blend of scientific rigor, perseverance in the face of daunting chemical challenges, and a collective determination to bring a promising laboratory observation to a point of clinical utility. Their efforts, meticulously documented and driven by an urgent sense of purpose as World War II loomed, would soon transform penicillin from a scientific curiosity into the world's first life-saving antibiotic, forever changing the landscape of medicine and offering humanity a powerful new weapon against infectious disease. With the compelling success of their animal trials, the Oxford team, led by Florey, Chain, and Heatley, turned their attention to the ultimate test, administering penicillin to human patients. The year was 1941, and the world was embroiled in the escalating turmoil of World War II, a conflict that underscored the urgent need for effective treatments for battlefield infections. The first human subject for penicillin treatment was Constable Albert Alexander, a police officer suffering from a severe staphylococcal and streptococcal infection that had developed from a scratch on his face. The infection had spread, leading to abscesses on his face and lungs, and he was gravely ill, his prognosis dire, with traditional treatments having failed completely. On February 12, 1941, Florey and his team administered the first dose of penicillin to Constable Alexander. The initial response was nothing short of miraculous. Within days, Alexander's condition showed dramatic improvement. His fever subsided, his appetite returned, and the infections began to recede, a truly astonishing turnaround for a man who had been at death's door. However, the Oxford team faced a formidable challenge: the extreme scarcity of penicillin. Their purification methods, while effective, yielded only minuscule quantities of the drug, and even with their ingenious methods of recycling penicillin from the patient's urine, they simply could not produce enough to sustain continuous treatment. After 5 days of remarkable progress, their precious supply of penicillin ran out. Tragically, without continued treatment, Constable Alexander's infection flared up again, and he eventually succumbed to his illness. This heartbreaking outcome underscored both the incredible potential and the immense practical hurdles that still needed to be overcome. Despite this setback, the initial, dramatic improvement in Constable Alexander provided crucial, undeniable proof of concept. Penicillin could indeed combat severe human infections. The team pressed on, conducting further trials on other patients, often those with life-threatening infections where all other treatments had failed. Subsequent patients, including a 15-year-old boy suffering from osteomyelitis and a young girl with a severe streptococcal infection, also showed remarkable responses to the limited doses they received. While some of these early patients also died due to the insufficient supply of the drug, the consistent pattern of initial improvement solidified the Oxford team's conviction that they were on the cusp of a medical revolution. These early human trials, though fraught with challenges and tinged with tragedy, provided the irrefutable clinical evidence necessary to propel penicillin from the laboratory into the realm of practical medicine. They demonstrated not only penicillin's potent antibacterial activity, but also its astonishing safety profile in humans, laying the groundwork for its eventual mass production and widespread use. The glimmer of hope sparked by Constable Alexander's brief recovery would soon illuminate the path for a new era in medicine, transforming the fight against infectious diseases and forever altering the landscape of human health and longevity. The urgent need for a sufficient supply of this miracle drug now became the paramount challenge, one that would require international collaboration and unprecedented industrial effort. As World War II raged across the globe, the desperate need for effective treatments for battlefield injuries, particularly infected wounds, became a critical imperative for the Allied forces. The Oxford team, having demonstrated penicillin's profound efficacy in their initial human trials, understood that their limited laboratory production methods were utterly insufficient to meet the looming demand. Recognizing the dire urgency and the potential to save countless lives, Howard Florey and Norman Heatley embarked on a crucial mission to the United States in the summer of 1941. Great Britain, under the constant threat of German bombing and with its industrial capacity fully dedicated to the war effort, lacked the resources and infrastructure for the large-scale fermentation and chemical processing required for penicillin production. In the United States, with its vast industrial capabilities and burgeoning pharmaceutical industry, lay the hope for scaling up production. Florey and Heatley tirelessly presented their findings to American scientists, pharmaceutical executives, and government officials, emphasizing penicillin's miraculous potential to combat infections that were crippling troops and civilians alike. Their compelling data, particularly the dramatic results from the human trials, convinced key figures of penicillin's strategic importance. The U.S. government, recognizing penicillin as a vital weapon in the war against infection, initiated an unprecedented collaboration between government agencies, universities, and private pharmaceutical companies. This collective effort, driven by wartime necessity, transcended traditional competitive boundaries, uniting scientific minds and industrial power in a singular, urgent mission. Researchers across the nation worked feverishly to find more efficient methods for producing penicillin, searching for higher-yielding strains of the Penicillium mold and developing innovative fermentation techniques. One significant breakthrough came with the discovery of Penicillium chrysogenum on a moldy cantaloupe in Peoria, Illinois, a strain that produced significantly higher quantities of penicillin than Fleming's original Penicillium notatum. Simultaneously, chemical engineers and industrial chemists tirelessly worked to optimize the extraction and purification processes, moving from small laboratory flasks to massive fermentation tanks. The challenges were immense. Maintaining sterility in large vats, optimizing nutrient media, and overcoming the chemical instability of penicillin. Companies like Merck, Pfizer, Squibb, and Abbott, driven by a blend of patriotism and humanitarianism, dedicated enormous resources to the project, overcoming engineering hurdles and pioneering new manufacturing techniques. By 1943, penicillin production had ramped up significantly, and by D-Day in June 1944, enough penicillin was available to treat all Allied casualties, dramatically reducing mortality rates from infected wounds and saving countless lives on the battlefield. The war effort served as an extraordinary catalyst, accelerating the development and mass production of penicillin in a way that would have been unimaginable in peacetime. This unprecedented collaboration between science, industry, and government fundamentally transformed the pharmaceutical landscape and established a new paradigm for drug development, demonstrating the immense power of collective action in the face of a global crisis. Penicillin, a product of accidental discovery and relentless scientific pursuit, emerged from the crucible of war as a symbol of hope and a testament to humanity's capacity for innovation under duress. The widespread availability of penicillin during and immediately after World War II ushered in a profound and unprecedented transformation in medicine, heralding the age of antibiotics and fundamentally altering the landscape of human health. What was once a death sentence, a simple bacterial infection, could now be treated effectively, often with dramatic speed and success. Diseases that had plagued humanity for millennia, silently claiming countless lives, suddenly met their match. Pneumonia, previously a leading cause of death, especially among the elderly and infirm, became a treatable condition, its once grim prognosis replaced by a high rate of recovery. Meningitis, a devastating inflammation of the brain and spinal cord, which had left survivors with severe neurological damage or claimed their lives swiftly, became conquerable. Syphilis and gonorrhea, sexually transmitted infections that caused long-term debilitating effects and often led to death, were now curable with a simple course of penicillin, revolutionizing public health efforts and significantly reducing the societal burden of these diseases. Childbirth, historically fraught with the peril of puerperal fever, became significantly safer for mothers, dramatically reducing maternal mortality rates and ensuring healthier outcomes for newborns. The impact on surgery was equally revolutionary. Operations that were once too risky due to the almost inevitable onset of post-operative infection could now be performed with greater confidence. Surgeons could delve deeper, perform more complex procedures, and save limbs and lives that would have been lost to sepsis just a few years prior. Amputations, once a common response to severe limb infections, became less frequent as penicillin could often arrest the spread of bacteria. The average life expectancy in developed nations saw a dramatic increase, a direct consequence of the newfound ability to combat infectious diseases that had previously cut lives short. Hospitals, once feared as places where infections were contracted rather than cured, began to transform into true centers of healing, where patients could recover from illnesses that would have been fatal only a generation before. The very concept of illness and recovery changed. No longer was a bacterial infection a matter of grim inevitability, but a challenge that could be met and overcome with targeted medical intervention. Penicillin also paved the way for the discovery and development of other antibiotics, sparking an intense period of antimicrobial research that would yield a diverse arsenal of drugs, each targeting different types of bacteria. The penicillin miracle not only saved millions of lives directly, but also laid the foundation for modern medicine as we know it, enabling advancements in surgery, transplantation, and cancer therapy, all of which rely on the ability to control bacterial infections. It transformed humanity's relationship with disease, shifting the balance of power from the microscopic world to human ingenuity, offering a profound sense of hope and control that had been absent for all of recorded history. The once invisible enemies were now visible, and, crucially, vulnerable, thanks to a chance encounter in a forgotten petri dish. The profound impact of penicillin on human health and its pivotal role in the Allied victory during World War II could not be ignored. The scientific community, though initially slow to embrace Fleming's findings, now universally acknowledged the revolutionary significance of the mold juice. In 1945, the Nobel Prize in Physiology or Medicine was jointly awarded to Sir Alexander Fleming, Sir Howard Florey, and Ernst Chain, recognizing their indispensable contributions to the discovery and development of penicillin. This shared honor acknowledged the distinct yet interconnected roles each played in this medical triumph. Fleming was honored for his original, serendipitous observation of penicillin's antibacterial properties, the crucial first spark of discovery that illuminated its potential. His keen eye and willingness to investigate an anomaly set the entire chain of events in motion. Florey and Chain were recognized for their tireless work in transforming that initial observation into a viable therapeutic agent. Florey's leadership, vision, and relentless pursuit of clinical application, combined with Chane's brilliant chemical expertise in purifying and understanding the compound, were essential in making penicillin a practical reality. While Norman Heatley, whose ingenious engineering and practical solutions were undeniably critical to the Oxford team's success, was not included in the Nobel Prize, his immense contributions are widely recognized by historians of science and remain an integral part of the penicillin story. The Nobel Prize solidified penicillin's place in history as a watershed moment, a testament to the power of scientific inquiry and collaboration. Break time equals 0. 25s/Beyond the accolades. The legacy of Fleming, Florey, and Chane extended far beyond the immediate saving of lives. Their work catalyzed the entire field of antibiotic research, inspiring countless scientists to search for new antimicrobial compounds, leading to the discovery of streptomycin, tetracyclines, and many other life-saving drugs. Penicillin became a global symbol of scientific progress and the humanitarian potential of medicine. Its success fundamentally changed the way pharmaceutical companies approach drug development, shifting focus towards rigorous scientific investigation and large-scale production. It also highlighted the critical importance of public health initiatives and the need for global cooperation in combating infectious diseases. Alexander Fleming, though a reluctant public figure, remained humble about his discovery, famously stating, "I did not invent penicillin. Nature did. I only discovered it." His humility underscored the accidental nature of his initial observation, yet it in no way diminished the brilliance of his insight and the perseverance of the Oxford team who brought it to fruition. The legacy of penicillin is not merely a tale of scientific achievement, but a testament to humanity's enduring quest to overcome suffering, a story of how a seemingly insignificant mold, through the hands and minds of dedicated scientists, reshaped the course of human health and offered a powerful weapon against unseen microbial adversaries, forever altering our relationship with disease and mortality. For years after its discovery and widespread use, the precise mechanism by which penicillin exerted its potent antibacterial effects remained a subject of intense scientific inquiry. While its ability to kill bacteria was undeniable, the how was a fascinating biological puzzle. Eventually, through meticulous biochemical and microbiological research, scientists unveiled penicillin's ingenious mode of action. It selectively targets and disrupts the synthesis of bacterial cell walls, a structure crucial for bacterial survival, while leaving human cells largely unharmed. Bacterial cell walls are rigid, protective layers that encapsulate the cell membrane, providing structural integrity and protecting the bacterium from osmotic lysis, the bursting of the cell due to water influx. These cell walls are composed primarily of peptidoglycan, a complex polymer unique to bacteria. The synthesis of peptidoglycan involves a series of enzymatic reactions, culminating in the cross-linking of peptide chains by a group of enzymes known as penicillin-binding proteins, PBPs, which are transpeptidases. Penicillin, remarkably, mimics the structure of the natural substrates that these PBPs bind to. It acts as a suicide inhibitor, irreversibly binding to the active site of the PBPs. By binding to these essential enzymes, penicillin effectively inactivates them, preventing the cross-linking of peptidoglycan strands. Without proper cross-linking, the bacterial cell wall becomes weak, porous, and structurally compromised. As the bacterium attempts to grow and divide, its internal osmotic pressure, which is significantly higher than its external environment, causes water to rush into the cell. Lacking a robust and intact cell wall to counteract this pressure, the bacterial cell swells and ultimately ruptures, leading to its death. This mechanism explains penicillin's bactericidal, bacteria-killing, rather than merely bacteriostatic, bacteria-inhibiting, effect. The genius of penicillin lies in its selective toxicity. Human cells do not possess peptidoglycan cell walls. Instead, animal cells have cell membranes, which are structurally and biochemically distinct from bacterial cell walls. Therefore, penicillin does not interfere with human cellular processes, explaining its remarkably low toxicity to the host. This precise molecular targeting was a revelation, providing a fundamental understanding of how antibiotics could selectively combat pathogens without harming the patient. Unveiling penicillin's mechanism of action not only deepened the understanding of bacterial biology, but also paved the way for the rational design of new antibiotics. Scientists could now search for other compounds that interfered with bacterial-specific processes, leading to the development of a diverse array of antimicrobial drugs, each with its unique target and spectrum of activity. This understanding underscored the elegant simplicity and profound effectiveness of nature's own antimicrobial strategies, adapted and harnessed by human ingenuity to wage war against invisible adversaries at a molecular level. The widespread and often indiscriminate use of penicillin and subsequent antibiotics, while revolutionary in saving countless lives, inadvertently set the stage for one of the most pressing public health crises of the 21st century: antibiotic resistance. Bacteria, with their rapid reproduction rates and remarkable genetic adaptability, possess an inherent capacity to evolve. When exposed to antibiotics, a selective pressure is applied. Susceptible bacteria are killed, while any naturally resistant variants survive and multiply, passing on their resistance genes to their progeny. This evolutionary process, often accelerated by misuse and overuse of antibiotics in both human medicine and agriculture, has led to the emergence of superbugs, bacterial strains that are resistant to multiple antibiotics, making infections increasingly difficult and sometimes impossible to treat. The mechanisms of resistance are varied and ingenious. Some bacteria develop enzymes, like beta-lactamases, that chemically break down and inactivate penicillin and related antibiotics. Others modify the target sites on their cell walls, preventing the antibiotic from binding effectively. Still others develop efflux pumps that actively pump the antibiotic out of the bacterial cell before it can exert its effect, or alter their metabolic pathways to bypass the block process. The problem is exacerbated by the inappropriate prescribing of antibiotics for viral infections, where they are ineffective, and by patients failing to complete their full course of antibiotics, allowing stronger, partially treated bacteria to survive and develop resistance. The widespread use of antibiotics in livestock, often to promote growth rather than treat illness, also contributes significantly to the pool of resistant bacteria, which can then transfer to humans through the food chain or environmental contact. The consequences of antibiotic resistance are dire. Common infections that were once easily treatable, such as pneumonia, urinary tract infections, and surgical site infections, are becoming increasingly difficult and expensive to manage, leading to longer hospital stays, increased healthcare costs, and, tragically, rising mortality rates. Surgical procedures, organ transplantation, chemotherapy for cancer, and the care of premature infants, all of which rely heavily on the ability to prevent and treat bacterial infections, are now threatened by the diminishing effectiveness of our antibiotic arsenal. Stopwatch equals 0.25s. Scientists and public health officials worldwide are sounding the alarm, warning that without urgent global action, we risk entering a post-antibiotic era where common infections and minor injuries could once again become life-threatening, effectively reversing decades of medical progress. Stopwatch equals 0.25s. The story of penicillin, while a testament to human ingenuity, also serves as a cautionary tale. The very tools that saved humanity now face an existential threat, demanding responsible stewardship, innovative research, and global collaboration to preserve their efficacy for future generations. Stopwatch equals 0.25s. The fight against infectious disease is an ongoing evolutionary arms race, and humanity must adapt and innovate to stay ahead of the relentless march of bacterial resistance. Stopwatch equals 0.25s. The escalating crisis of antibiotic resistance has spurred a renewed and urgent global effort to discover and develop new antimicrobial compounds, pushing the frontiers of scientific research into innovative and often unexpected directions. Recognizing that the traditional low-hanging fruit of easily discoverable antibiotics has largely been picked, scientists are now employing sophisticated technologies and exploring novel biological pathways to identify the next generation of life-saving drugs. One promising avenue involves delving deeper into natural environments, exploring untapped reservoirs of microbial diversity. Researchers are meticulously sifting through soil samples from extreme environments, marine sediments, and even the human microbiome, searching for microorganisms that produce unique defensive compounds. Many bacteria and fungi naturally compete with each other, producing a vast array of antimicrobial molecules, some of which may hold the key to overcoming current resistance mechanisms. Beyond traditional natural product screening, advanced genomic sequencing technologies are allowing scientists to rapidly analyze microbial DNA, identifying genes responsible for producing novel antimicrobial peptides or secondary metabolites, even if the compounds themselves are not easily culturable in the laboratory. This genomics-driven discovery bypasses the limitations of traditional cultivation methods, unlocking a vast potential pharmacopoeia. Stopwatch equals 0.25s. Synthetic biology and medicinal chemistry are also playing crucial roles, with researchers designing entirely new molecules from scratch, specifically engineered to evade bacterial resistance mechanisms or target previously unexploited bacterial vulnerabilities. This includes developing compounds that inhibit bacterial virulence factors, preventing them from causing harm even if they are not directly killed, or molecules that can disarm resistance genes. Phage therapy, a century-old concept, is experiencing a remarkable resurgence. Stopwatch equals 0.25s. Bacteriophages, viruses that specifically infect and kill bacteria, offer a highly targeted approach to combating resistant infections, particularly in cases where conventional antibiotics have failed. Stopwatch equals 0.25s. Researchers are also investigating combination therapies, using existing antibiotics in novel combinations to enhance their efficacy or overcome resistance, and exploring adjuvants that can restore the susceptibility of resistant bacteria to older drugs. Stopwatch equals 0.25s. Immunomodulatory approaches, which boost the host's own immune response to better fight off infections, represent another exciting frontier, moving beyond directly killing bacteria to empowering the body's natural defenses. Stopwatch equals 0.25s. The development of rapid diagnostic tests is also critical. Quickly identifying the specific pathogen and its resistance profile allows for targeted antibiotic use, reducing the indiscriminate prescribing that fuels resistance. Stopwatch equals 0.25s. The challenges are immense, from the high costs of drug development to the complexities of clinical trials, but the ongoing quest for new antimicrobials is a testament to humanity's unwavering commitment to health and the recognition that the fight against infectious disease is a continuous and evolving battle. Stopwatch equals 0.25s. The next penicillin miracle may not be an accidental discovery, but the product of concerted, multidisciplinary scientific ingenuity, safeguarding the gains achieved over the past century and ensuring a future where infections remain treatable. Stopwatch equals 1s. The enduring efficacy of antibiotics, including penicillin, hinges not only on the discovery of new drugs, but, crucially, on the responsible stewardship of the existing arsenal and a concerted global approach to public health. The battle against antibiotic resistance is not one that can be won by individual nations or isolated scientific endeavors. It demands a unified, comprehensive strategy spanning medical practice, agriculture, research, and policy. Responsible stewardship begins at the point of prescription. Clinicians must exercise prudence, prescribing antibiotics only when necessary, for the correct duration, and targeting the specific pathogen whenever possible, rather than using broad-spectrum drugs indiscriminately. Public education campaigns are vital to raise awareness among patients about the appropriate use of antibiotics, dispelling misconceptions, and discouraging self-medication or the demand for antibiotics for viral illnesses. Implementing robust infection prevention and control measures in hospitals and healthcare settings is paramount to break chains of transmission for resistant bacteria reducing the need for antibiotics in the first place. This includes stringent hand hygiene, proper sanitation, and the judicious use of isolation protocols. In agriculture, reducing the routine use of antibiotics for growth promotion in livestock and transitioning towards healthier animal husbandry practices is a critical step in mitigating the spread of resistance in the food chain and environment. Surveillance programs are essential to monitor the emergence and spread of resistant strains, providing real-time data to inform public health interventions and guide the development of new treatments. This requires robust laboratory capacity and data-sharing mechanisms across borders. Global health initiatives are indispensable because bacteria do not respect national boundaries. International collaboration is needed for research and development of new antibiotics, vaccines, and rapid diagnostics, addressing the economic disincentives that often hinder pharmaceutical investment in this area. Developing equitable access to existing and future antibiotics is also crucial, as limited access in some regions can lead to suboptimal treatment and foster resistance, while overuse in others accelerates the problem. Stopwatch equals 0.25s, the One Health approach, recognizing the interconnectedness of human, animal, and environmental health, is increasingly vital, acknowledging that antimicrobial resistance is a complex issue requiring holistic solutions across these sectors. Stopwatch equals 0.25s. Investing in basic science to understand bacterial evolution and mechanisms of resistance, and fostering innovative approaches to drug discovery, remains a long-term imperative. Stopwatch equals 0.25s. The legacy of penicillin demands not only celebration but also vigilant protection. Stopwatch equals 0.25s. The miracle drug that saved humanity is now under threat from within, from the very forces of evolution it helped to unleash. Stopwatch equals 0.25s. Responsible stewardship is not merely a medical or scientific concern. It is a collective societal responsibility, a moral imperative to preserve one of humanity's greatest scientific achievements for generations to come, ensuring that the promise of treatable infections remains a reality. Stopwatch equals 0.25s. The choices made today will determine the health landscape of tomorrow. The story of penicillin is far more than a mere scientific narrative. It is a profound reflection on humanity's enduring journey, a testament to our capacity for discovery, resilience, and occasional serendipity in the face of relentless natural challenges. From the constant, ancient fear of unseen microbial invaders that shaped human civilization for millennia to the dramatic transformation brought about by a single, accidental observation, penicillin encapsulates the very essence of human progress. It reminds us that monumental breakthroughs often begin with humble beginnings, sometimes a forgotten petri dish, and that the greatest triumphs can emerge from unexpected places, revealing the extraordinary within the ordinary. The quiet genius of Alexander Fleming, the tireless dedication of the Oxford team, and the unprecedented industrial effort during wartime illustrate the power of individual insight coupled with collective action, a synergy that can overcome seemingly insurmountable obstacles. Penicillin not only saved countless lives, allowing generations to live longer, healthier, and more productive lives, but it also fundamentally reshaped our understanding of disease and the possibilities of medical intervention. It liberated humanity from the constant dread of common infections, enabling advancements in surgery, public health, and chronic disease management that would have been unthinkable in the pre-antibiotic era. Yet, the very success of penicillin also unveiled a deeper truth about the dynamic and complex relationship between humanity and the natural world. That every triumph can bring new challenges, and that progress is not a final destination, but a continuous process of adaptation, innovation, and stewardship. The emergence of antibiotic resistance serves as a powerful reminder of nature's relentless evolutionary capacity and the delicate balance we must maintain in our interventions. It underscores the responsibility that comes with great power and the need for humility and foresight in wielding the tools of science. The ongoing quest for new antimicrobials, the vigilant efforts in antibiotic stewardship, and the commitment to global health collaboration are not just scientific imperatives. They are reflections of humanity's ongoing commitment to a healthier future, a testament to our collective will to continue the fight against suffering. The legacy of penicillin is etched into the fabric of modern life, visible in every hospital, every successful surgery, every recovered patient. It is a story of hope, of resilience, and of the profound impact one discovery, accidentally stumbled upon, can have on the entire human species. As we look to the future, the lessons learned from penicillin's journey, the importance of observation, perseverance, collaboration, and responsible stewardship remain guiding principles in our ongoing journey to understand and navigate the intricate dance between life and disease. The accidental discovery of penicillin stands as an eternal beacon, illuminating humanity's capacity to transcend perceived limitations and to continue the endless journey of unraveling the mysteries of the universe, one microscopic breakthrough at a time. The narrative of penicillin, from a forgotten petri dish to a global savior, offers a profound and resonant reflection on the intertwined destinies of humanity and the microscopic world. For millennia, humanity existed in a precarious balance with unseen microbial adversaries, their lives often truncated by infections that defied understanding and treatment. Diseases like pneumonia, tuberculosis, and countless wound infections were not just ailments, but silent, pervasive architects of mortality, dictating the very rhythms of life and limiting the human experience. Then, in a moment of extraordinary serendipity and astute observation, Alexander Fleming, a quiet bacteriologist, stumbled upon a mold that held the key to unlocking this ancient biological prison. His keen eye, distinguishing a mere contamination from a profound biological interaction, ignited a chain of events that would forever alter the course of medicine. The subsequent tireless efforts of the Oxford team, led by Florey, Chain, and Heatley, transformed Fleming's laboratory curiosity into a revolutionary drug, a feat of scientific ingenuity and collaboration achieved under the immense pressure of global conflict. Penicillin's impact was immediate and transformative, saving millions of lives during World War II and beyond, ushering in the age of antibiotics and fundamentally reshaping our relationship with disease and death. It became a symbol of hope, a testament to humanity's capacity to overcome seemingly insurmountable challenges through scientific inquiry and collective action. Yet, the story does not end with triumphant success. It evolves into a cautionary tale, a poignant reminder that scientific advancements, however miraculous, are not static victories. The rise of antibiotic resistance, an inevitable consequence of evolution accelerated by human practices, underscores the dynamic and ongoing nature of the battle against infectious disease. It challenges us to reflect on our responsibility as stewards of these invaluable medical tools, urging us towards prudent use, continuous innovation, and global cooperation. The journey of penicillin, therefore, is a microcosm of humanity's larger scientific endeavor, a continuous cycle of discovery, adaptation, and the relentless pursuit of knowledge to improve the human condition. It stands as an enduring monument to the power of a single idea, the collaborative spirit of science, and the profound, often unexpected, ways in which a moment of accidental discovery can reverberate through time, shaping the destiny of civilizations and offering a brighter, healthier future for all. The lessons from penicillin resonate deeply today, inspiring us to remain vigilant, to innovate tirelessly, and to cherish the delicate balance between scientific triumph and ecological responsibility in our ongoing quest for health and longevity.